intron, the 3' end. So in the 3' splice site, in general, this is recognized by a dimer, the U2AF dimer, that recognizes together the AG at the end of the intron and a polypyrimidine tract. So happens that in yeast, this is missing. So even though the branch site is recognized, this factor is missing. So the question comes, how is this correct AG selected in yeast? So that's the topic of my talk. So how is 3' selection, 3' splice site selection in yeast going on? So there are two problems. One is that you may find other AGs between the real exons and the branch site. So how the splicing machinery knows which one is the correct AG. And also, in about a third of the introns in yeast, doesn't have many, but a third of them, which is about 100, the distance between the branch site and the splice site is quite long, like too long to really explain by the simple models of the splicing. So there must be some kind of scanning going on or a way to get the right AG. So the literature was enough to think of this hypothesis that secondary structures may help in this recognition. The idea is the following. So cryptic AGs, the ones that are not real splice sites, will be hidden by base pairing with other positions in the intron. And at the same time, this will help the approach the branch site to the correct splice site. Okay, so we wanted to see whether this is a valid hypothesis. So we looked at the introns, and we can separate the introns into the ones that have a structure and those that don't have a structure. And those that have a structure, if you take the optimal structure, or even if you do it with a suboptimal structure, the same thing happens. So if you cut out the structure and you calculate the effective distance, so you can prove that the effective distances are similar to the distances in the introns without a structure. So you may say, okay, but this happens to any sequence. The longer the sequence you take, there is always a structure. But the maximum effective distance is significantly different. So real introns tend to approach more branch site and 3' splice site than random sequences. Okay. The other hypothesis is that how structures may hide the AGs, which are cryptic. So to do this, what we did is scan the region between the branch site and the 3' splice site with windows of different length. For every window, you calculate possible structures. And for every AG that you find in these windows, you summarize the base pair probability. This is a summary measure of how likely is this AG to be occurring in a base pairing situation. When you do this, what you find out is that real 3' splice sites, in general, they are more accessible than cryptic ones which occur in the intron or in the exon. So accessibility is basically 1 minus the probability of being in a base pair. And we see here that real splice sites are more accessible. Okay, so it seems to make sense hypothesis. If you do this with other Saccharomyces, you see the same trend. Real splice sites have higher accessibility. And also if you consider structures in the introns and you remove the structure and calculate the effective distance, this also seems to approach the branch site to the 3' splice site. So our experimental collaborators validated this hypothesis by basically mutating a predicted structure. So we don't know whether this is real, but there might be one because of the computational prediction. So we mutate the bases so that we destroy the base pairing. And then what they see in this case is that the splice site now is 2 is not chosen anymore, and the 1 is chosen instead. If they recover the structure by doing compensatory mutations, they recover back the splice site 2. And if they basically remove the structure but also delete the AG, since this AG now is far away from the branch site, so the splicing is not occurring anymore. So the structure seems to be compatible with the recognition of the splice site. Okay, so maybe since my experimental collaborators are not here, I would say that an experimentalist would say, okay, fine, we have a model, and then publish. That's fine, but what I would like to see is that whether that is really general. With this hypothesis, can you do a mathematical or some kind of predictive model and try to prove that this is correct for all the or most of the introns. So the idea is that taking these sequence properties, can we explain the selection of most of the 3' splice sites, and can we also use this to predict alternative sites? So what we did is build a support vector machine. That is basically a classifier that can distinguish real from non-real 3' splice sites. And to do this, we use a training set, the annotated, and all the cryptic is AGs, which are not annotated as 3' splice sites. And for those cases, we collect some information, like what actual sequence it is, like is this a CAG or TAG, the distance to the branch site, accessibility measure, as we calculated before, pyrimidine contact, and the distance to the pyrimidine. And okay, since there are more negative cases than positive, what we do is to subsample 200 cases to have a balanced training subset. And with this, that means that we sample a number of times, and each time we sample, we train a support vector machine. In fact, we do 10,000 times, and every time you predict on the remaining set, and we generate a score, which is basically the number of times, or the proportion of times, in this case, in 10,000 models, that a given possible AG, a given AG is classified correctly as negative or as positive. So that will be our score. So our model is really like a family of 10,000 different models trained after subsampling. So if we do that, we find a very good precision, very good sensitivity. So we predict with a false positive rate of around 3%, we predict 92% of the annotated 3' splice sites. That's the AUC. So, but one thing that we like to see, and I actually don't see very much in people describing predictive models, is a precision recall curve, because this is telling us that our precision is really bad. That means that to be able to achieve this sensitivity, you have to predict lots of false positives. In fact, we have to predict many, many cases which are wrongly predicted to be able to see all these correct cases. So we have to still improve this. Nevertheless, the score of our prediction correlates very well with the number of reads or the number of cases that are covered by deep sequencing reads. These deep sequencing reads were taken from published papers where they did RNA sequencing on yeast. Okay, so we are on a good track, but we still need to improve this. Let me point out that the features that are the best separators for this model are actually not the accessibility, which is related to the secondary structure, but in fact it's the distance to the branch site and the pyrimidine content. But it's still accessibility counts in also for the prediction. Okay, so we can improve the precision. This is also interesting from the point of view of predicting alternative sites. What does it mean to predict an alternative site? Because the whole point is that since yeast is so well annotated or so exhaustively annotated, there is no point in building a predictive model for 3' splice site in yeast. These are all known. With the model, we actually, it's a proof of concept that the secondary structure can explain most of the annotated 3' splice site, but we already know which ones are these. But let's say if we try to use it to predict alternative 3' splice sites, what do we have to do? So basically, the alternative ones would be those which they look very much like the annotated ones, but they are false in general because they're not annotated in the database. So that means that these are our false positives, right, in our set. So if we take the false positive from our prediction, those are candidates for alternative sites. However, we know that to be more reliable for those, we have to reduce the false positive rate. What I mean is, if we are able to predict very few false positives, but those are very likely to be like the annotated ones, maybe those are alternative ones. That was our approach to predict alternative ones. But to do that, we have to modify the threshold. So that is, in each of the SVMs, we have a default threshold that's 0. So anything that above 0 is positive, below 0 is negative. But if we modify the threshold so that every time we predict, we predict with a very low false positive rate, like less than 0.5%, then what we're doing is increase the precision of the model. And that's indeed the case. So this was the previous model, and now with the new way of scoring, what we're doing basically is reduce sensitivity, so we predict less annotated cases. We're still benchmarking here on the annotated cases, doing M-fold cross-validation, but we have much better precision. That means that, even with this scoring system, very few will be false positives, but maybe those are more likely to be alternative sites. And still the score correlates with the coverage by deep sequencing reads. Okay, so we selected the best 33 cases predicted as alternative sites, and then we went on to validate them. So remember, these are things which are predicted as false positives, but since our model is built at such a low false positive rate, then we think they are the best candidates. Okay, so this is one of the tested cases. So this was one prediction. Here in the picture, I show all the cryptic AGs along the intron and the exon, downstream exon. This is the coverage of RNA sequencing reads. These are the reads spanning the exon-exon junctions. So for this, so the secondary structure corresponding to this region is depicted here. So the color means red is the pair probability. So if it's in a base pairing position, means like highly likely to be in a base pairing position. If it's outside, it's highly likely not to be in a base pairing position. So that means that we have this quite a strong base pairing, but this is quite a strong not in a base pair. So this is alternative one, which is the intronic one. And so our collaborators use different yeast strains to validate this case. For this case, they have to use primer specific RT-PCR because if they use primers on the two exons, the level of inclusion of this alternative site is so low that you won't see the alternative splicing. But if you have specific primers for the junction, then you're able to detect the alternative sites. So this will be the 3' and the alternative, sorry, this is the alternative site. I don't know why, yeah, this one here, sorry. We also predicted on a completely independent set. So in yeast, there are also 5' UTR introns, very few, but there are some, and these were not included in our training set. So we took those introns and tried to first validate our model with the annotated splice sites, and we predicted 91% of them correctly. And we also predicted 4 alternative ones. These are the 4 ones. Some of them also had some RNA sequencing reads. And we validated the case of the MTR2. And in this case, this is weird because that's the way they annotate 5' UTR introns in UCSC, but there are reads spanning this alternative site as well, and we predicted this to be also forming a secondary structure approaching the 2 alternative sites, and this is the validation on different yeast strains. Okay, so this seems to be general, and then the hypothesis that this may also help in finding alternative sites could be correct as well. So finally, the secondary structure, bear in mind that this is dependent on temperature. So the temperature may also vary the predictions, and therefore we have to take this into account. So we predicted all our tests were done 22 degrees, but under heat shock conditions in yeast, so of course the structural predictions change, and some of our predictions also changed. So this is a case where there was an alternative 3' splice site, which is now downstream. That means this is exonic, and the structure is inside the exon. So the optimal structure is the same one for both cases. That's okay because it's just one optimal structure. That means that's the most probable one. But what changes is the accessibility and the base pair probability of all the bases. If you can see, all orange of this CAG goes into more red CAG for the alternative sites at high temperature. That means that the accessibility increases for the alternative site. So our model doesn't predict this one at 22 degrees, but it predicts this one at 37. However, when we validated this experimentally, we can see it at different temperatures. So this is probably simply because our model is a ranking. What we do is to take a ranking. So this is was below our threshold for the top cases that we use for prediction, but it seems that it's also real for 22 degrees. Okay, so here you can see that the validation by RT-PCR of this is a real one, this is the alternative one, and these are the controls. So there is no signal in the controls, and so the rate between the inclusion of both sites changes between temperatures. So there is a higher inclusion, different rate of inclusion between the two temperatures. So there seems to be also a role played by the temperature in this case. Okay, so what I've tried to explain to you is that we start from a hypothesis that we want to explain the 3' selection in yeast. There was enough evidence to hypothesize that secondary structure may play a role, and then by doing genome-wide analysis is consistent with the hypothesis, and experimental tests also are consistent with the hypothesis, and then making a predictive model seems also to be able to describe this, not only the recognition of 3' splice sites, but also the possibility that there is alternative splicing in yeast. Okay, so with this I'd like to finish acknowledging that this is mostly the work by one of my PhD students now. Is a postdoc in Copenhagen, and this was a collaboration with Pedro Gabriel Ferreira in CRG and Josep Vilardell also in another lab in Barcelona. Okay, thank you.
Did you try to run your models on other yeasts or Pombe?
Okay, so Pombe has the U2AF dimer. So the main issue is that if there are structures, may behave completely different. So this hypothesis won't work. For the other yeast, we were relying on the fact that our experimental collaborators were working with the cerevisiae yeast species. So, but all the data looked like other Saccharomyces could work exactly the same way.
You mentioned that you used the minimum free energy structures for all your experiments, right? But that you also tried them on suboptimal structures. Can you elaborate a little bit more on that, and specifically if you have seen any direct consequences by using the suboptimal structures?
Yeah, so I don't have the plot with me, but the whole point is this graph. So this is basically showing that if you take the optimal structure and remove it and calculate the effective distance, you come back to a distance which is a distribution like in the introns without structure. But if you do this with shuffled introns or with random sequences, you have the same behavior. The difference is that the maximum effective distance is longer and significantly longer for shuffled introns and random sequences. So also, if you, instead of taking one single structure, what you do is to sample structures per intron. So you do this not just using one structure per intron, but multiple structures, also suboptimal ones. You still see that the distribution of all these suboptimal structures are significantly different from the shuffled or random sequences.
You did it also calculate the energies for 22 degrees Celsius and 37. You calculated the energies for 22 degrees Celsius and 37 degrees Celsius. But the parameters are originally for 37, and we had the experience that the downscaled ones are actually not as good for the 22 or lower degrees than maybe sometimes using the normal parameters which have been experimentally validated. So do you have problems with the scaling of the parameters to different temperatures?
I'm not sure. So there's the parameters of the model, you mean?
The parameters of the Turner model have been determined at 37 degrees Celsius.
Oh, yes.
If you fold at 22 degrees, it's just predicted parameters or calculated parameters, which are not experimentally validated. And then often there you introduce errors by using this. Have you seen this kind of effects?
I'm not sure I can hear you well, the question. So I didn't hear you well. So we did the model, we trained the model at 22 and 37 degrees.
So you fold at 22 with RNA fold or something like that, right? So
we do.
The parameters
we
are
do
only experimentally validated for 37 degrees Celsius.
Okay. Yeah,
for 22, these are calculated parameters.
Okay, I see.
Which are not necessary. So for loop energy and stack energy.
Okay. Yes.
Which are not maybe right. So have you seen effects of that?
Yeah, so one effect would be the fact that we cannot predict this last example. So one effect is the following: we didn't see very many differences. So we were expecting much more differences in the prediction at 22 and 37. So if I get the table, so this table is actually in the paper, includes whether the alternative site is predicted at both temperatures, and most of them are predicted at both temperatures. This, to me, doesn't make much sense. So you should see more differences. So that's probably what's going on. Yes.